For centuries, the way we've understood life, really, has been dominated by this one image, the cell, as just a biochemical machine.
Right, like a little bag of chemicals following rules.
Exactly. Deterministic molecular collisions, genetic blueprints dictating everything. It's powerful, sure, but maybe a bit simplistic.
Well, yeah, it definitely feels reductionist. It sort of strips the cell down, makes it passive almost. It misses that spark of intelligence, of self-organization you see everywhere in life.
And then that's what we're exploring today. We're kind of setting that old blueprint aside to dive deep into a really different way of looking at things. This comes from the Unified Coherence Theory of Everything, the UCTE. And its core idea is pretty fascinating. Cells aren't machines. They're actually resonant, intelligent agents.
That's the shift. And our sources for this deep dive, mainly Chapter 8, Cells as Resonant Intelligent Agents, and Chapter 9, Tissues as Resonance Networks, they give us a really detailed, structured look at this. We're basically moving beyond just chemistry here. We're looking at life through the lens of coherence and resonance. So, our mission today is to really unpack the details, the architecture of this theory, so you can see how profound the implications might be.
Okay, so before we get into the nuts and bolts, we need some new language, right? This theory has a couple of key terms that change how we think about biological information.
That's right. Okay, let's unpack this, starting with the smallest, most fundamental unit, the basic unit of information transmission. They call it the hologene.
The hologene. Okay, so if a traditional gene is the blueprint for, say, a protein,
Yeah.
what's the hologene doing?
So the hologene isn't a molecule in the same way. It's the basic transmissive unit of cellular coherence. Think of it more like a specific frequency pattern. A bioelectric code the cell projects.
Like a signal.
Exactly, like a single pure note in a huge orchestra carrying very specific information. Not about making something necessarily, but about a potential state, a potential identity or function for the cell.
Okay, so the cell sends out these hologenes, these patterns, and when all those individual signals from countless cells come together, they integrate.
Yes, they synchronize, they integrate into a larger field, and that integrated whole, that's the hologene.
Ah, the hologene.
That's the collective systemic coherence, the field of a whole tissue or an organ or, the entire organism. If the hologene is the cell's individual note, the hologene is the whole symphony. It's the emergent, unified field that organizes everything, maybe even consciousness itself, built from all those cellular outputs.
Wow. Okay, so we're shifting from thinking about information stored in molecules, the gene, to information carried in frequencies and fields. That's the hologene and hologene. That's our foundation there.
That's the groundwork, yeah.
All right, so section 1, saying goodbye to purely mechanistic biology.
Well, the big critique from the UCTE perspective is that the old mechanistic model just can't explain the sheer speed, the efficiency, the adaptability of life.
How so?
I mean, if cells only relied on slow, random molecules bumping into each other, how could an organism possibly organize itself, react quickly enough to survive all the constant pressures from the environment? It seems too slow, too clumsy.
Yeah, the traditional view makes the cell seem kind of passive, right? Just reacting to chemicals, which then begs the question, if the cells are just chemical reactors, where does the organism's intelligence, its ability to act purposefully, actually come from?
And the UCTE answers that by saying the cell isn't passive at all. It reframes the cell as a kind of micro-observer, an active participant that can actually select its state and contribute to the bigger picture, the systemic coherence. It's not just waiting for orders.
Okay, so what gives the cell this agency? What makes it more than a machine? The UCTE points to 3 key properties, right?
That's right. 3 properties that define it as an intelligent agent. First is coherence orientation.
Coherence orientation.
This is its built-in fundamental ability to maintain internal order. To fight against entropy, the tendency towards disorder, we call that negentropy.
So it actively maintains itself.
Exactly. A machine breaks down, it needs outside help. A living cell has this internal drive to stay organized, to heal, to persist.
Okay, that makes sense. What's the second property?
The second is resonance communication. This goes way beyond just chemicals crossing a synapse. It's how cells talk to each other almost instantaneously.
Yeah.
By exchanging those hologenes we talked about through bioelectric fields that travel incredibly fast, near light speed, communication here is about matching frequencies, aligning phases, not just molecules fitting into receptors. It's much faster, more holistic.
Wow, okay. Instantaneous communication. And the third property? This one sounds like the big one.
It is pretty provocative. It's adaptive selection. This is the cell's ability to actually choose its state of coherence.
Yeah.
It uses what the theory calls observer function dynamics. Basically, faced with possibilities, the cell selects one. It decides.
Hang on, that sounds potentially chaotic. If you have billions of cells all deciding things independently, wouldn't the whole system just fall apart? How does agency on that scale not lead to total breakdown?
But that's the crucial point the UCTE addresses. The cell's agency isn't random or selfish. It's always fundamentally in service to the whole. It's an intelligent agent, yes, but a cooperative one.
How is that cooperation maintained?
Its selection process is guided by strict criteria that prioritize the overall systemic order above all else. It can read the resonance inputs, figure out the best state for the whole system, and then project that choice back out. It's an agent, but an agent of coherence intelligence.
Okay, so for the cell to do all this complex stuff, communicate instantly, adapt, select, it needs the right internal machinery, an integrated system. And this is the cellular coherence triad.
Exactly. The triad consists of three key players working together: DNA, microtubules, and the cell membranes. They function as this integrated coherence engine.
Right. Let's start with DNA. We think we know DNA, the blueprint for proteins, but UCTE sees it differently.
Fundamentally differently. It's redefined as the instructional resonator. So yes, it holds information, but it's not just a static blueprint. It's seen as a spectral library of hologenes. Its primary role isn't just chemical instructions, it's transmitting resonance patterns.
Patterns. Like frequencies again?
Yes, specific frequencies that define the vast potential identities and states the cell could adopt. It's the library containing all the possible songs the cell could possibly play. It sets the potential.
Got it. So DNA provides the library of potential resonances. Then, what actually takes those patterns and turns them into action, into the dynamic flow within the cell? That's the microtubules.
That's where the microtubules come in. They function as the quantum executors.
Quantum executors. That sounds intense.
Well, it involves some pretty fascinating physics. These microtubules, they're tiny hollow cylinders running throughout the cell's cytoplasm. They are the highways for this coherence information.
And the sources get specific about how they do this, right? How do they manage these coherence flows so well?
They do. We need to slow down a bit here because the details are amazing. First mechanism, soliton transmission. Yeah, a soliton is a special kind of wave. It's self-reinforcing. It travels without losing its shape or energy. Think of sending a perfectly stable pulse down a rope. It doesn't just fade away. Microtubules use these solitons to send coherent information packets across the cell without degradation.
Wow, so perfect information transfer, basically. No data loss. That's crucial for maintaining that order, the entropy. What else helps?
Well, they don't exist in a vacuum. They're surrounded by the cell's internal environment, which isn't just plain water. It's highly structured water. The UCTE calls it aquahel buffering.
Structured water.
Yeah, it's described as a specific phase of water, almost like liquid crystal or ice-like layers that form around the microtubules.
Okay, kind of like insulation.
Or like an organic fiber optic cable. That's a good analogy. The structured water does two things. It helps insulate the signal, minimizing energy loss, and it actually helps align the microtubule and thus the cell with the larger systemic resonance fields, the hologene.
Okay, so it keeps the signal clean and keeps the cell tuned into the bigger network. And there was a quantum aspect too, tunneling currents.
Yes. This points to the quantum mechanical nature of the process. It suggests electrons can tunnel, essentially jump instantaneously across energy barriers within the microtubule structure that they couldn't cross classically.
So it's not just plumbing, it's a quantum conduit.
Exactly. Microtubules bridge the potential resonance information from the DNA to the cell's outer boundary, making the connection between instruction and action, potential, and actuality.
Which brings us to that outer boundary, the final piece of the triad, the membranes as resonance interfaces.
Right. The cell membrane isn't just a passive wall keeping things in or out. It's incredibly active. It's the cell's antenna system, both a projector and a receiver for those hologene signals.
And its electrical properties are key.
Absolutely crucial. The voltage gradients across the membrane, the constant electrical oscillations, these aren't just byproducts, they are fundamental. They embed the cell within the larger tissue and organism-level coherence fields. They're how the cell broadcasts its status and receives signals from the network.
So the membrane voltage is like the cell's status update. Its way of saying, here I am, this is what I'm doing, to the rest of the body.
That's a really good way to put it. The whole triad DNA holding the potential patterns, microtubules executing the resonance flow quantum mechanically, and the membrane interfacing with the wider network, it works as one sophisticated engine. It keeps the cell coherent internally while ensuring it's fully plugged into the organism's living field.
Okay, this is where it gets really interesting for me. We've talked about the hardware, the triad. Now, how does the cell use it? How does this intelligence actually show up moment to moment? You mentioned decision-making.
Yes, we move from just reactions to the cell as a decision node.
A decision node.
Think about it. Any single cell, at any given instant, is just a wash in information. It's receiving countless resonance potentials from inside and out.
What kind of potentials?
Oh, possibilities for everything. Which genes to express, what metabolic pathway to use, whether to divide, repair itself, communicate something specific, even whether to undergo programmed cell death, apoptosis. It's a huge field of options.
And instead of just automatically reacting based on the strongest chemical signal.
Exactly. Instead of a purely mechanical, pre-programmed response, the UCTE says the cell performs coherence selection.
Coherence selection. That's the core of its agency, then.
Yeah.
It actively narrows down all those possibilities into the one state it actually adopts. How should we picture that?
Well, the analogy used is quite helpful. Think of tuning an old analog radio. The airwaves are full of stations, right? Thousands of signals, all those resonance potentials. The radio doesn't play static by trying to process all of them. It tunes itself. It adjusts its internal circuits to resonate with one specific frequency, one station, and that's the song you hear. It collapses the field of potential broadcasts into one actualized output. The cell does something like that with hologenes. It tunes into one potential state.
And the source material draws a parallel here to quantum physics, doesn't it? The observer effect.
It does. What's fascinating here is this process mirrors the observer function in physics. The act of observation, in quantum mechanics, collapses the wave function from a state of potentiality into a definite state. Here, the cell is the observer, and its act of selection collapses the biological possibility field into biological reality.
But it's not just picking randomly, is it? There must be rules, criteria for this selection.
Oh, absolutely. It's not random at all. It's governed by a deep contextual intelligence. The cell has to essentially answer 3 crucial questions simultaneously to make the right choice for the system.
Okay, what's the first criterion?
First off, there's the internal focus, internal resonance stability. Basically, which choice, which potential state best helps the cell maintain its own internal order, its own entropy, which state maximizes its own coherence and chances of survival.
Right. Makes sense. The cell has to look after itself first, to some extent, but you said it's cooperative.
Exactly. So that internal drive gets balanced by the second criterion, systemic alignment.
Aligning with the whole system.
Precisely. Which potential state aligns best with the overall hologenetic coherence, the needs of the tissue, the organ, the entire organism. The cell has to choose the option that contributes positively to the whole symphony, even if it means, say, limiting its own growth or performing a specific, maybe strenuous function for the collective. This is the key to preventing chaos.
Okay, self-preservation, balance with system needs. What's the third layer?
The third layer is looking outward, environmental feedback. The cell has to select the state that resonates appropriately with what's going on outside the organism, too. Things like external stresses, signals from other organisms nearby, nutrient levels, even electromagnetic fields in the environment. It's a dynamic balancing act. Self, system, and environment.
Wow, that's complex decision-making at the cellular level. Can we see this playing out in real biological examples?
Definitely. The theory provides some compelling examples. Think about stem cell differentiation. That's maybe the ultimate coherence selection event.
How so?
Well, a stem cell starts out pluripotent. It holds the potential resonance patterns, the hologenes, for becoming many different types of cells. Its decision to become, say, a specific neuron or a bone cell is a massive act of collapsing that potential. It selects one functional hologene out of countless possibilities and commits to that role, aligning with the needs of the developing tissue.
Okay, that's a great example. What else?
Consider immune recognition. When a lymphocyte, an immune cell, encounters something, its recognition of self versus non-self, like a virus or bacteria, is incredibly fast and specific. The UCTE proposes this isn't just about molecular shapes fitting together, it's a high-speed resonance matching process. The lymphocyte is rapidly performing coherent selection. Does the signal resonate as self or danger? It's a coherence-based decision.
Faster than chemical binding alone could explain.
Potentially, yes. And think about healing responses.
Yeah.
When you get a cut, fibroblasts move into the area to repair the tissue. According to this theory, they aren't just responding passively to chemical growth factors, they are actively selecting specific regeneration hologenes. They're tuning into the resonance patterns needed to restore the local coherence field of the damaged tissue, ensuring it aligns with the systemic hologene template of what that tissue should look like. These are context-dependent, intelligent choices.
This really paints the cell as an active partner, doesn't it? Not just a cog in the machine.
Exactly. It leads to what the theory calls the observer-participant model. The cell isn't just being observed or instructed, it's participating in a constant feedback loop.
Can you walk us through that, Luke?
Sure. The cell receives hologenes information from its environment and the systemic field, input. It then performs coherence selection based on those three criteria we discussed: internal stability, systemic alignment, environmental feedback selection. Then, crucially, it projects its own updated hologene pattern back out via its membrane field projection. And finally, the organism's overall hologene field constantly integrates all these projections from billions of cells, updating the state of the whole system. Integration. It's a continuous, dynamic, participatory dance. The cell is actively co-creating the organism's reality moment by moment.
So I've got the individual cell as this resonant, intelligent agent making decisions. But life isn't just individual cells, it's tissues, organs, the whole organism working together with incredible efficiency. How does this scale up? How do we get collective cellular intelligence?
That's the next critical step. How do billions of these individual observer participants synchronize to create something far greater than the sum of its parts, and do it almost instantaneously?
Yeah, it sounds like a coordination nightmare if it were just chemical signals.
It probably would be. But the UCTE suggests it happens through field-level coherence mechanisms, largely bypassing slower chemical routes. One key mechanism is gap junctions.
Okay, what are those?
They're actual physical channels connecting adjacent cells directly, tiny tunnels. They allow ions and electrical signals to pass directly from one cell's interior to the next.
So they're literally wired together electrically.
In a way, yes. It ensures immediate electrical and ionic synchronicity in local neighborhoods of cells, like plugging instruments directly into the same timing clock.
Okay, so that handles local sync. What about coordinating larger areas, whole tissues?
Then you have voltage gradients. Remember those electrical potentials across cell membranes. Across a whole tissue, these align and create larger scale voltage fields. These act like bioelectric maps, guiding development, regeneration, and maintaining tissue-wide resonance patterns.
Like an electrical blueprint for the tissue's form and function.
Sort of, yes. And perhaps most importantly, there's field projection. Every single cell, by projecting its hologenes out through its membrane, isn't just communicating its own state. It's actively contributing to and shaping the shared systemic field, the hologene. It's a collective broadcast that creates the overall coherence.
So when we see something amazing like morphogenesis, how an embryo shapes itself into complex forms or how tissues regenerate after injury, this isn't just genes switching on and off in sequence.
According to UCTE, no. It's guided by these shared synchronized coherence fields. The bioelectric fields act as guiding templates orchestrating the cellular activities. Genes are involved, of course, providing the potential patterns, the hologenes, but the field dynamics coordinate the expression in space and time.
Okay, and all these collective cellular projections from all the tissues and organs together,
Yeah.
that creates the organism's overall field.
Yes, that collective resonance projection creates the organism-wide bioelectric field. The UCTE calls this simply the biofield. And importantly, it's not seen as just some fuzzy energy byproduct of metabolism. It's defined as the systemic coherence envelope of the organism. It's the living, resonant template that holds the information for the organism's form and function.
Right, and this leads us down a pretty radical path regarding consciousness, doesn't it? If every cell is an observer participant contributing to this field, then the old idea that consciousness lives only in the brain.
It starts to look incomplete, doesn't it? The source material makes a bold claim here. Consciousness is likely distributed across all cells.
Distributed consciousness.
Yes. Neurons, in this view, are highly specialized cells. They act as amplifiers, processors, and integrators of these coherent signals, playing a crucial role, obviously. But they might not be the sole source of consciousness. Consciousness could be the emergent property arising from the highest order integration of all those countless cellular observations within the unified hologene field.
The theory uses a specific term for this, a fractal cascade of observers.
That's the one. It paints a picture of nested levels of observation and integration. You have the individual cell observing and selecting its state. Then the tissue synchronizes these selections into a local field. Then the organ integrates multiple tissue fields. And finally, the unified organism, the systemic hologene, achieves the highest level of integration which we experience as consciousness.
So intelligence and awareness aren't just top down from the brain, but built up from the bottom, from every cell contributing.
That's the implication. It's an integrated intelligence spanning the entire body. And this framework, interestingly, even offers a potential scientific lens for concepts like the aura.
Ah, right. Often dismissed as purely mystical. How does UCTE approach that?
It reframes the traditional idea of the aura in terms of measurable physics. It suggests the aura corresponds to resonance shells. These are the layered external projections of the systemic biofield, actual electromagnetic fields extending beyond the skin that, in principle, could be measured.
And because they are resonant structures.
Because they are holographic coherent structures, they wouldn't just reflect the organism's present state. They would also contain information about its history, its resonance memory, like ripples on a pond preserving the pattern of past disturbances.
So the aura, in this view, is literally the radiating field, carrying the integrated coherence history and current state of the organism.
Oh.
That connects something seemingly esoteric right back to biophysics.
It attempts to tie it all together, suggesting consciousness arises from the whole body's integrated cellular intelligence, projecting a field that carries its state and memory.
Okay, so we've gone from the single-cell intelligence up to the whole organism's biofield and distributed consciousness. Now let's get back down to the physical structure. How are tissues actually built to handle these different resonance functions? The idea is that they aren't just clumps of cells, but specialized resonance networks, each with a unique functional tissue code.
Exactly.
Here's where it gets really interesting. When we break down the fundamental building blocks of the body by their unique resonance signature.
Right, and this specialization, these different codes, aren't arbitrary. The UCTE argues they derive directly from fundamental principles of spatial organization, how biological structures arrange themselves in 3D space using three key geometric ideas: chirality, torsion, and curvature.
Okay, let's quickly define those so we're all on the same page.
Sure. Chirality is basically handedness. Think of your left and right hands. They're mirror images, but you can't perfectly superimpose them. In biology, this shows up in things like the direction a helix winds, left or right. It dictates a bias and flow, whether it's information or force.
Okay, handedness. What about torsion?
Torsion is about twisting or spiraling. Think of wringing out a towel. This twisting motion is crucial in biology for concentrating energy, generating force, or creating specific types of waves or signals like those solitons we mentioned.
Got it. Twist. And curvature.
Curvature is simply about bending or folding, how surfaces curve in space. This allows tissues to create pockets, channels, wells areas where resonance can be focused, concentrated, contained, or directed.
Chirality, torsion, curvature, handedness, twist, bend. These three geometric tools shape the four main tissue types and give them their specific resonance jobs. Let's break them down. First up, neural tissue information coherence.
Neural tissue is obviously optimized for handling information rapid transmission, complex integration. Its resonance signature is high-frequency oscillations and very fast synchronization, what's called phase locking.
And how did the geometric principles show up here? Chirality.
Chirality is clear in the neuron structure. The axon transmits signals one way. Dendrites receive, that's a fundamental polarity, a directional bias for information flow.
Makes sense. And torsion and curvature in neurons.
Torsion is thought to be involved in how those soliton waves propagate down the axon, a twisting, spiraling transmission of the coherent signal. And curvature is really pronounced at the junctions, the synaptic terminals and the dendritic spines. They form these highly curved structures that act like little coherence wells, concentrating the signal right at the point of transmission or reception, maximizing efficiency and speed.
Okay, so neural tissue uses geometry for fast, directed information flow. Next, muscle tissue mechanical coherence, the movers.
Right, muscle is all about converting resonance patterns into physical force, into directed work. Its signature is mid-frequency oscillations and powerful torsional contractile waves. It generates force through twisting motions.
Where's the geometry? Chirality.
Chirality is fundamental in the structure of the contractile filaments themselves. The actin and myosin helices wind in specific directions. This inherent handedness biases the direction of the contraction and relaxation cycle.
And torsion seems obvious here.
Oh, absolutely dominant. Torsion is muscle contraction, essentially. The way the myosin heads bind to actin and pull isn't just a linear tug, it's a spiraling, twisting motion, the cross-bridge cycle. This generates directed force very effectively, and curvature is seen in how the basic units, the sarcomeres, are folded and packed, allowing for the efficient chemical and electrical energy release needed for powerful, coordinated movement.
Makes sense. Torsion for force. Now, the tissue that holds everything together and provides the environment, connective tissue stability, coherence.
Yes, the connective tissue network, fascia, tendons, ligaments, bone matrix, it's the structural resonance ground layer for the whole body. It's often overlooked, but absolutely crucial. Its signature is low-frequency, long-wavelength oscillations. It's designed for stability, integration, and a key property, piezoelectric responsiveness.
Piezoelectric. Can you explain that clearly for us? What does that mean in the body?
It means that when you apply a mechanical stress to connective tissue, stretching it, compressing it, shearing it through movement, or even just posture under gravity, the tissue structure itself directly converts that mechanical energy into an electrical signal, a coherence signal.
Wow. So the entire connective tissue network is like a giant sensor, translating physical forces into electrical information.
Exactly. It's a whole-body mechanosensor, constantly translating the physical reality of movement and structure into the language of coherence.
And its structure is built for this. How does geometry play in?
Perfectly. Chirality is seen in the collagen fibers themselves. They're typically triple helices, winding in a specific way. This structure is incredibly strong and influences how forces are transmitted along the fiber network. This allows the fascia sheets to handle torsional shear forces effectively. And the way these sheets curve and fold, curvature, creates a continuum, distributing mechanical loads and resonance signals over large areas, acting as coherence wells that store and dissipate energy slowly. It provides the stable, low-frequency background hum that the faster systems like nerves need to function properly.
Incredible. The body's living fabric translating mechanics to electricity.
Yeah.
Okay. Fourth and final code. Epithelial tissue interface. Coherence. The boundaries.
Epithelial tissues are the linings and covering skin, the lining of the gut, lungs, glands. They are the critical boundary operators. They manage the coherence exchange between the organism's internal environment and the external world. Their resonance is characterized by polarity-driven fields, allowing for dual projection, managing what comes in and what goes out.
How does geometry feature here?
Chirality. Chirality is clearly expressed in the fundamental apical-basal polarity of epithelial cells. The top surface, facing the lumen or outside world, is different from the bottom surface facing the internal tissues. This creates different flow biases for absorption versus secretion, managing coherence exchange directionally.
Right, and torsion and curvature in shaping these boundaries.
Torsion becomes really important during development, during morphogenesis. Epithelial sheets often undergo complex twisting and folding torsion to form tubes, ducts, and glands, creating specific resonance channels. And curvature is obviously essential as these sheets fold to create structures like the alveoli in the lungs or the villi in the intestines. These folds create bounded resonance wells where specific functions like gas exchange or nutrient absorption can occur efficiently, concentrating the necessary coherence fields.
So epithelial tissues are like the organism's resonant skin, shielding, sensing the outside, projecting the inside, managing the flow between the two. It's amazing how these four distinct tissue codes, built on simple geometric principles, create such specialized resonance functions.
It really paints a picture of the body as a sophisticated polyphonic resonance system.
Okay, so we have these specialized tissues with their unique resonance codes. How did they come together to form functional organs?
Well, tissues provide the specialized notes, but organs are where the harmony happens. Organs are nested hologenes. They emerge when multiple tissue codes, neural, muscle, connective, epithelial, integrate harmonically into a higher-order functional unit.
And this coherence isn't just local to the organ, right? It's layered.
Exactly. It's nested.
Yeah.
There's the local coherence field within the organ itself, defining its specific function. But that organ-level hologene is also nested within and contributing to the systemic hologene of the whole organism. And the UCTE even suggests this nesting extends outward, with the organism potentially resonating with larger environmental or even cosmic fields. It's coherence at multiple scales.
And this combination of tissue codes leads to different organ types or archetypes based on which code is dominant.
Precisely. We can see clear organ archetypes.
For example, The brain is obviously the primary neural dominant archetype. Its main job is high-frequency information coherence, integrating all those signals from the fractal cascade of observers we talked about. It's the master processor.
Right. Then you have the heart, which is powerfully muscle dominant. The UCTE describes it as the body's primary torsional resonance engine. It doesn't just pump blood, it generates a huge electromagnetic field and drives a rhythmic coherence throughout the entire system.
A torsional engine, I like that. What about the lungs?
The lungs are strongly epithelial dominant. They are critical boundary regulators, managing that vital coherence exchange between the inside and outside, not just gases, but potential environmental frequencies too.
Yeah, and something like the liver. It seems more complex.
The liver is a great example of a connective epithelial hybrid. It has a massive connective tissue structure providing stability and complex epithelial arrangements for filtering and processing. Functionally, it acts as a key resonance stabilizer and filter for the whole system, maintaining the balance of the systemic hologene. Even organs like the kidneys are defined by their highly specialized curvature-dominant epithelial structures, forming intricate resonance wells designed for ultrafiltration.
So when you put all these specialized organs, these nested hologenes, together, integrating all the tissue codes across the whole body, you get the complete picture, the organism as a resonance architecture.
That's the term used, or perhaps a more evocative image from the sources is the organism as a resonance cathedral.
The resonance cathedral.
Yeah, a dynamic living structure built from light and frequency, where every level of coherence, from the quantum events and microtubules all the way up to the whole body biofield, integrates into this unified, systemic, holographic hologene. It's an architecture of coherence, not just an assembly of parts.
So what does this all mean? It means your body is less like a car needing a chemical tune-up.
And maybe infinitely more like a complex symphony that needs all its instruments to be in tune and playing in phase.
Which brings us to health and disease. If the body is a resonance architecture,
then health and disease have to be understood in terms of resonance. Health is simply the state where this complex resonance architecture is maintained, where all the parts are coherent, synchronized, resonating harmoniously with the overall hologene.
And disease.
Disease is dissonance. It's what happens when parts of the system fall out of phase, when coherence codes get disrupted or degraded. This creates destructive interference patterns, noise in the system, which eventually manifests as structural breakdown or functional impairment.
So logically then, healing is resonance restoration.
Exactly. Healing is the process of coherence repair. It involves reestablishing the correct resonance patterns, guided by the inherent templates within the hologene. The goal is to bring the dissonant parts back into alignment, back into phase with the entire resonant hierarchy, restoring harmony to the symphony. So, just to recap the core ideas from this really dense but fascinating UCTE framework we've explored. Fundamentally, the cell isn't a passive machine, it's an observer agent. It actively performs coherence selection moment by moment. Life communicates and organizes itself using resonant codes, the hologenes at the cellular level, integrating into the systemic hologene. This collective resonance creates the organism-wide biofield. And consciousness isn't just in the brain, it's likely distributed, emerging from a fractal cascade of nested observers throughout the body. The body itself is a resonance architecture built from specialized tissue codes, neural, muscle, connective, epithelial, based on fundamental geometry, all converging into this living resonant whole.
It really shifts the perspective. If life is fundamentally participatory, if consciousness emerges from the collective coherence of all our cells, then everything we do for restoration, moving our bodies, reducing stress, engaging in healing practices, isn't just tweaking chemistry. We're actively participating in the coherence refinement of our own biofield, maybe even the universal field. We're literally tuning our own resonance shells.
It gives you a different sense of your own agency, doesn't it? Knowing that at a cellular level, you're constantly involved in collapsing potential into the reality of your physical being.
It absolutely does. And it leaves us with a pretty profound, provocative question to ponder. Are we just passive observers watching our reality unfold? Or are we, through the constant collective coherence selection happening in billions of intelligent cells every second, actually selecting it? Are we projecting our coherence outward and actively building the architecture of our lives? Something to think about next time you feel a little out of tune or perfectly in sync.